Hello everybody, this is Dr. Nadeem and we are with Neelam Path Lectures, the Pursue series. As you are aware, all our lectures are available on YouTube. We also have a Telegram group which is very useful for accessing all lecture related information. You can join the group. We also have a Google Drive where the PDF of all the lectures are uploaded and the master integration key which navigates between the PDF and the YouTube. These are the disclaimers, and we are in phase 3, which is the recorded pathology lectures. And today we have Pursue 22F, which is neoplasia, and we are streaming live from Peerless Hospital, Kolkata. And to talk on today's topic, we have Dr. Bhaskar Narayan Chaudhuri. He is the Chief Microbiologist and Senior Consultant in the Department of Microbiology and Molecular Biology and Infection Control Officer of the Peerless Hospital and Research Center, Kolkata. He has published and presented 88 papers and articles in national and international journals and in national and international conferences. He is the NABL lead assessor and technical assessor in microbiology, serology, and molecular testing, flow cytometry for medical laboratories. He is NABL-nominated trainer for ISO 15189-2022 training program of NABL. He is an advisor microbiology and molecular biology in the Medinova Diagnostics, Kolkata. Dr. Bhaskar Narayan Chaudhuri shall be speaking on microbiological etiology of cancers. So with much ado, I would like to call upon Dr. Bhaskar Narayan Chaudhuri to start his lecture on microbiological etiology of cancers. Sir, Dr. Bhaskar Narayan Chaudhuri, all yours. Thank you.
Hello everyone, I am Dr. Bhaskar Narayan Chaudhuri, Chief Microbiologist and Senior Consultant, Department of Microbiology and Molecular Biology, Peerless Hospital, Kolkata. My topic today is microbial etiology of cancers. So, what is oncogenesis? Oncogenesis is a process by which cancer-causing agents convert a normal cell into a cancer cell. So there is a proto-oncogene in the DNA, that is human DNA, which gets converted by these cancer-causing agents into activated oncogene and converts a normal cell into a cancer cell. So there are many tumor antigens like the oncofetal antigen CEA, which is involved in bowel cancer, AP involved in germ cell tumors, hepatocellular cancer, other antigens like CA125 in ovarian cancer, MUC1 breast cancer, and so on. But we have the DNA viral determinants and the RNA viral determinants. So these are microbial viral determinants which may cause cancer. So classes of human tumor antigens recognized by T lymphocytes with their genetic processes. You can see here there is high tumoral specificity of oncogenic viruses. So oncogenic viruses can affect all types of normal cells and can convert the normal cells into tumor cells. So these are known as what are the viral etiology of cancer. I'm coming to that first. So oncogenic viruses. So there is proto-oncogenesis which converts into normal cell into a cancer cell. So there are tumor suppressor genes which usually suppress this process, but when these tumor suppressor genes are deactivated by some cancer causing agents, then proto-oncogenesis occurs and proto-oncogene is converted into oncogene. So oncogenesis is the result of genetic changes that alter the expression or function of proteins that play critical roles in the control of cell growth and division. So oncogenic viruses cause cancer by inducing changes that affect cell growth and division. So what are the tumor viruses? Many animal cancers are retroviral in origin, so they are caused by retroviruses. So transmissible single-stranded RNA gets converted into double-stranded DNA by the means of reverse transcriptase enzyme, which is present in these viruses. Then it is integrated into the host cell genome. If remobilized, and it will carry the host cell DNA containing growth promoting gene, and it becomes potentially transforming. So that is what is known as a viral oncogene. Example, HTLV1 tax protein in the PX region of the viral genome activates growth promoting genes. So microbes inducing cancers not only include the viruses, although the viruses are the most important which can cause cancers. So they include the DNA viruses. I am mainly speaking on human cancers, so I will focus my talk on human cancer causing agents only. So DNA viruses include human papilloma virus, hepatitis B virus, the herpes viruses like herpes simplex virus 2, Epstein-Barr virus, and human herpes virus type 8. Molluscum contagiosum virus MCV, which is a pox virus causing Merkel cell carcinoma, polyona, and adenocarcinoma in animals, but it is not proven to be a cancer causing virus in humans so far. And amongst the RNA viruses, we have the hepatitis C virus and the retro-oncovirus, that is the HTLV1 and 2 viruses. Not only viruses, we have also bacteria causing cancers like Helicobacter pylori and Streptococcus bovis. Fungal aflatoxin aspergillus, which is found in Aspergillus flavus, may be associated in cancer. And amongst the parasites, trematodes like Clonorchis sinensis, Opisthochis flelinensis, Opisthochis viverrini, and Cystosoma hematobium have cancer-causing potentials. So coming to the viral etiology first. Human papilloma virus or HPV, it has got more than 66 types. It has been detected in oral and tonsil cancer tissues. So venereal transmission usually occurs, it's sexually transmitted and causes CA cervix. So CA cervix associated with mainly human papilloma virus 16 and 18. Others are 31, 45, 51, 52 and 53. These may also cause carcinoma of cervix. It may also cause CIN, that is cervical intraepithelial neoplasia, intraepithelial dysplasia of anus, particularly HPV 6, 11 and some other types are involved in this. Genital warts, condyloma acuminata and laryngeal papillomatosis are benign conditions but benign tumors which may be caused by HPV 6 and 11. Uncomplicated lower genitourinary tract infection and condylomas atypia of cervix can progress to CIN, that is cervical intraepithelial neoplasia, and it has got 3 grades: low grade SIL, that is squamous intraepithelial lesion, high grade SIL or carcinoma in situ, which is a precancerous condition. So HPV is involved in all these different types of HPV. So this is CA cervix and the throat warts, which may also be caused by HPV, and it is also a vaccine preventable cancer. So CA cervix can be prevented by the HPV vaccine. So this HPV vaccine is a bit controversial, but the best time to give it is at the age of 12 to 13 years, just at the start of adolescence, that is at the age of 13 years, in both girls and boys, particularly the girls, for the best effect. But it can be given up to 40 years of age. So according to the American Chemical Society, it recommends cervical cancer screening by HPV test every 5 years for females with a cervix from the age 25 until the age of 65. However, if HPV test, that is HPV DNA PCR, if it is not available, screening with a PAP, with a HPV or PAP co-test every 5 years or a PAP smear test every 3 years, that is recommended. Cervical brushing fluid, tissue or oropharyngeal swab may be run for HPV DNA PCR by open system, or if it is also available in closed systems, in some closed systems also, but we are using mainly the open system. So detection and genotyping of 14 HPV high or medium risk types like 16, 18, 31, 33, 35, 39, 45, 51, 52 and so on, these can be done through PCR. So PCR is the best method. If PCR is not available, you have to do pap smear test every 3 years in females with a cervix from the age 25 until the age of 65. So that is the recommendation. Now E7 protein of HPV-16, it binds and inactivates tumor suppressor gene RB. So E6 protein of HPV-18, it has got sequence homology to SV40, a polyoma virus. So it's a large T antigen, it binds and inactivates tumor suppressor gene P53. Both are necessary and sufficient to cause cell transformation in vitro. So it may also occur in vivo. And HSV2, which is herpes simplex virus 2, acts as a cofactor in CS cervix. Now coming to Epstein-Barr virus. So it's a virus which is associated with leukemia, mainly chronic myeloid leukemia, ABL gene in chromosome 9q, named after Abelson-Murine leukemia virus, which is a retrovirus. So Epstein-Barr virus may cause nasopharyngeal carcinoma, which is endemic in the Mediterranean, Far East countries. So they have got high, nasopharyngeal cancer patients have got high antibody titers of anti-HBV antibody. Titers measured to screen high-risk population. So these titers can be measured to screen high-risk population. Anaplastic nasopharyngeal cancer tissues, they contain Epstein-Barr virus DNA and antigens. This EBV DNA can be recovered by PCR and antigens by serological tests. So acute lymphocytic leukemias may occur in children, young adults. So L3, that is Burkitt's lymphoma leukemia, may occur in developing countries in children and young adults. Particularly Central Africa, 90% of this Burkitt's lymphoma, and 15% of Burkitt lymphoma in USA are associated with Epstein-Barr virus infection in infancy. So mono or oligoclonal proliferation of Epstein-Barr virus infected cells in 20 to 40% patients of Hodgkin's disease occurs, especially mixed cellularity type. 50% cases, in 50% cases, viral DNA and antigens are found in Reed-Sternberg cells. Aggressive non-Hodgkin's lymphoma may occur in immunosuppressed patients in the West, and the causative agent is Epstein-Barr virus. There is a strong association of Epstein-Barr virus with extranodal anti-angiocentric nasal T NK cell lymphomas in Asia and South America. HIV infection increases the risk of Hodgkin's disease, aggressive B-cell NHL, that is non-Hodgkin's lymphoma, immunoblastic lymphomas, which 20% of them are primary CNS lymphomas. 50% of these cases of immunoblastic lymphomas demonstrate HBV DNA by PCR, including all CNS lymphomas. All the CNS lymphomas demonstrate HBV DNA. HBV DNA is also found in some cases of tonsillar cancer, T-cell lymphoma, thymoma, gastric carcinoma, CNS lymphoma in the immunocompetent patients, leiomyosarcomas in AIDS patients, smooth muscle tumors in organ transplant recipients. So these are the diseases associated with Epstein-Barr virus, and out of these many are cancers like Hodgkin's lymphoma, gastric cancer, nasopharyngeal cancers, Burkitt's lymphoma. Now coming to the next virus, DNA virus, which may cause cancer, that is the human herpes virus 8, type 8, which causes Kaposi sarcoma, particularly in AIDS patients or immunocompromised patients. So Kaposi sarcoma in HIV positive, HIV-1 positive patients, it includes classic Kaposi sarcoma, and Kaposi sarcoma in HIV negative homosexual men may also occur, and it's associated with HHV8. Primary effusion lymphoma or body cavity based lymphoma in HIV positive patients may also be caused by HHV8. Multiple myeloma infection of bone marrow macrophages which HHV8 occurs in some cases. Viral interleukin 6 may contribute to the pathogenesis. Now coming to the hepatitis viruses, one is hepatitis B virus which is a DNA virus, and hepatitis C virus which is a RNA virus. Both of them are oncogenic viruses. So chronic infection with hepatitis B or hepatitis C virus may lead to cirrhosis, and the risk of hepatocellular cancer in cirrhotic patients is at the rate of 3% per year. So these hepatitis viruses are the principal reason for high incidence of liver cancer in parts of Asia and Africa. Incidence is 100-fold higher than in normal population in the patients who harbor hepatitis B or hepatitis C viruses in their liver. Hepatitis B virus DNA may be integrated into the host genomic DNA in tumor cells and adjacent uninvolved liver cells. Modifications of cellular gene expression may occur by insertional mutagenesis, chromosomal rearrangements, transcriptional transactivating activities of X and pre-S2 regions of the HBV genome. So, what is oncogenesis? Oncogenesis is a process by which cancer-causing agents convert a normal cell into a cancer cell. So there is a proto-oncogene in the DNA, that is human DNA, which gets converted by these cancer-causing agents into activated oncogene and converts a normal cell into a cancer cell. HIV infection increases the risk of Hodgkin's disease, aggressive B cell NHL, that is non-Hodgkin's lymphoma, immunoblastic lymphomas, which 20% of them are primary CNS lymphomas. 50% of these cases of immunoblastic lymphomas demonstrate HBV DNA by PCR, including all CNS lymphomas. All the CNS lymphomas demonstrate HBV DNA. HBV DNA is also found in some cases of tonsillar cancer, T-cell lymphoma, thymoma, gastric carcinoma, CNS lymphoma in the immunocompetent patients, leiomyosarcomas in AIDS patients, smooth muscle tumors in organ transplant recipients. Now, hepatitis B vaccination can prevent hepatitis B virus-induced liver cancer. So, it is a vaccine-preventable cancer, just like cervical cancer, which can be prevented through HPV vaccine, which is a bit controversial, but there is no controversy regarding hepatitis B virus. Now, it is recommended for all persons, all the entire population, not only for the healthcare workers, where it is very important, but it is now recommended as an universal immunization program in India for all infants to be vaccinated. So Serum Institute of India guidelines for hepatitis B immunized high risk persons. So complete vaccination includes 3 doses at 0, 1 month and 6 months, and it gives protection for around 25 years. Seroconversion is implied by anti-HBs titer. It should be more than 10 IU per liter or 10 MIU per microliter. If it doesn't occur, this anti-HBs titer should be checked at least 6 to 8 weeks after giving the last dose of the vaccine. And if the titer is not more than 10 IU per liter, then we have to revaccinate that person and check the titer again after a whole course of revaccination. But again, after the second course of vaccination, around 10% of the population remain non-responders for life. It is advisable for healthcare workers in high-risk category to get anti-HBs titer checked every 5 to 10 years and get a booster dose if the titer falls below 10 IU per liter. Now coming through to the retrovirus, that is HTLV1. This is endemic in Southwest Japan, South Africa, West Africa, Caribbean, South Asia and South America. Transmission mainly occurs through transplacental route. It may also be transmitted from mother to the baby by breast milk, and also transmitted via blood transfusion or by the sexual route. It is associated with adult T-cell lymphoma or leukemia, that is patients acquiring virus from mother through breast milk. 2.5 to 4% of them become carriers, and there is a latency thereafter for around 10 to 55 years, and then they manifest this adult T-cell lymphoma leukemia. HTLV2, it was first isolated from a case of T-cell variant of hairy cell leukemia. Subsequently, it was not found to be consistently associated with any disease. Hence, HTLV2 is sometimes called a virus searching for a disease. HIV, which is also a retrovirus, is not an oncovirus, but it may be associated with cancers. Mostly is associated with lymphomas. So lymphomas is the commonest cancer that may occur in HIV affected individuals who turn into AIDS. And next is Kaposi's sarcoma, and there is twofold rise in the incidence of CS cervix, CIN, intraepithelial dysplasia of anus than in normal population in HIV infected persons. Antivirals as anti-cancer agents, it also proves that viruses act as have cancer causing potential. So antiviral interferon alpha and beta, they are used in chemotherapy of genital and other warts, which is a benign condition, papillomas intralesional, which is also a benign condition, and recurrent juvenile laryngeal papillomatosis caused by HPV, which is also a benign condition. But interferon alpha may be used in chemotherapy of Kaposi's sarcoma, hairy T-cell leukemia. Complete response occur in 50% of the patients of hairy T-cell leukemia, and also in hepatitis B and C carriers, interferon alpha 2A and 2B. Now coming to the bacteria, the most important bacteria is Helicobacter pylori. Nitrates in dried, smoked, salted foods may be converted to carcinogenic nitrites by bacteria introduced exogenously through partially decayed foods. These are usually consumed by lower socioeconomic classes and may cause adenocarcinoma of stomach. So Helicobacter pylori contributes to the above effect by causing chronic gastritis, loss of gastric acidity, bacterial growth in stomach, followed by metaplasia, cellular atypia, and then neoplasia, that is adenocarcinoma. So Helicobacter pylori has been found in as an etiologic agent of adenocarcinoma. So there is increased risk of gastric lymphomas also, especially mold, that is mucosa associated lymphoma, lymphoid tissues, malt lymphomas, mucosa associated lymphoid tissues, lymphomas, low-grade B-cell lymphomas also. Patients treated with anti-H. pylori regime, they show regression of malt lymphomas. Bacteria doesn't transform lymphocytes. A vigorous immune response to the bacteria may cause chronic antigenic stimulation leading to neoplasia. So H. pylori, there are many tests. The most important is the rapid urease test or urea breath test, through which we can diagnose H. pylori. And the best is best is PCR, or the biopsy from the stomach has to be sent for PCR or some other tests. Next bacteria is the Streptococcus bovis. subvariant of Streptococcus bovis is Streptococcus galolyticus. It's a subspecies. So Streptococcus bovis biotype 1, the new nomenclature of which is Streptococcus galolyticus. It's a group D non-enterococcal fecal streptococcus. It may cause endocarditis, bacteremia and septicemia, but for unknown reasons 60% of such patients have colorectal tumors, that is carcinoma of the colon, and also polyps, which is a benign condition, and sometimes upper GI cancers also may be caused by Streptococcus galolyticus, which is Streptococcus bovis biotype 1. Now coming to fungal etiology, aflatoxin beta 1, which is a mycotoxin of Aspergillus flavus infecting agricultural crops like corn, that is maize, peanuts, cotton seeds, etc. It's an important public health hazard in Africa and South China. It may induce a specific mutation at codon 249 in tumor suppressor gene P53 and may cause hepatoma in rats. Human hepatoma connection is still uncertain, but it may be involved in human hepatoma also. Cancer immunotherapy. So immunotherapy against cancer, some of the bacteria and can be used in cancer immunotherapy like BCG, Bacilli calmette-guerin, and non-living Corynebacterium parvum. They have got role in acute leukemias, melanoma and breast cancer. Glucan, a pyran compound present in fungal cell wall, they stimulate CMI, that is cell-mediated immunity, and macrophage functions, and may act against cancers. So they can also play a role in immunotherapy against cancers. Now coming to parasitic etiology. So parasites like the trematodes includes Schistosoma hematobium, which is associated with increased incidence of urinary bladder cancer, which is a squamous cell cancer. Squamous cell cancer in urinary bladder cancer, squamous cell origin may occur in 70% of the cases, and transitional cell origin in 30% of the cases. And this is common in many of the developing countries. So this Schistosoma hematobium causes bilharziosis, and this may lead to bladder cancer, both squamous cell in most of the cases and transitional cell cancers in 30% of the cases. Next is biliary flux, Clonorchis sinensis, Opisthorchis felinensis, Opisthorchis viverrini. They cause periductal or biliary inflammation leading to fibrosis, gallbladder becomes enlarged, and there is increased risk of cholecystitis, cholelithiasis and cholangiocarcinoma, and this may also lead to pancreatic carcinoma. So cholangiocarcinoma and pancreatic carcinoma may be caused by the biliary flux. Cholangiocarcinoma epidemiology related to Clonorchis sinensis infection in China and Opisthorchis viverrini infection in northeast Thailand. There is a strong association and many papers have been published on this. So cancer doesn't have a bleak future. We can't tell that cancer has a bleak future because we have got much advancements in cancer etiology, cancer treatment and cancer diagnostics and treatment. But if many of the cancers have microbial etiology, there is definitely a ray of hope. So that is the end of my presentation. Thank you.